Good morning, grade 9 students. This is Ms. Nidal. Today, we're going to continue the explanation of Chapter 7, Chromosomes Carriers of the Genetic Information, Activity 2, The Laws of Heredity. Let's first recall what we have already seen in the first activity. What is heredity? Heredity is the transmission of hereditary traits from a living organism to its offspring. To study the transmission of these traits, crosses are performed. Then, what is a monohybrid cross? It's a cross between two pure lines that differ from each other in one trait, so one gene is studied. Two cases will be discussed, the case of dominance and the case of codominance and incomplete dominance. So, let's start with the case of dominance. What is a monohybrid cross with dominance? It's a cross between two different pure lines where one gene is studied and one allele of this gene is dominant over the other allele, which is recessive. So please open your book and observe document A, page 136. Here is an example about the case of dominance. A cross-fertilization is realized between the pistil of flowers of smooth pea plants of pure line with the pollen of flowers of wrinkled pea plant of pure line. These pure lines form the parental generation or generation P. The seeds that resulted from this cross were all smooth and formed the first generation or F1. This F1 generation consists only of smooth seeds. This means that the allele smooth is dominant over the allele wrinkled, which is recessive. Then, the smooth seeds of F1 are crossed with each other, and this is what we call self-fertilization. The self-cross gives a second generation called F2 generation. As you can see in the figure, the F2 generation consists of 3/4 smooth seeds and 1/4 wrinkled seeds. All these results are experimental. Then we need to verify these experimental results. So, how to verify these experimental results? A factorial analysis is required. Since the allele smooth is dominant over the allele wrinkled, then smooth will be represented by a capital letter and wrinkled will be represented by a small letter. Let S be the symbol of the allele responsible for the trait smooth, dominant over its allele wrinkled. Let W be the symbol of the allele responsible for the trait wrinkled that is recessive. Parents. The first parent has the phenotype S and the second has the phenotype W. The genotypes are SS, WW, since these parents are pure line. SS produces 100% gamete S, so one type of gamete. Also, WW produces only one type of gamete, which is W. Thus, the first generation, resulting from the union of these two gametes, consists of SW, phenotype S, so 100% seeds with the phenotype smooth. When the self-cross is performed between F1, the phenotypes being S, the genotypes SW, being hybrids, then each one of them produces two types of gamete, half S, half W. The F2 generation resulting from this self-cross will consist then of SS 1/4, SW 1/4, SW 1/4, and WW 1/4. Thus, the phenotypic results obtained are S 3/4 and W 1/4, and we can say that these experimental results are verified. Let's move now to the test cross. What is a test cross? As we saw in the previous experiment, the phenotypic results obtained are 3/4 smooth and 1/4 wrinkled. Among the 3/4 smooth seeds, which is a dominant phenotype, some of them must be SS pure and others SW hybrids. So the question is, how can we identify the genotype of an individual with dominant phenotype, like for example, the smooth seeds? This is possible through performing a test cross. What is a test cross? It's when the individual with dominant phenotype, like S, of unknown genotype, is crossed with an individual of recessive phenotype, like for example, wrinkled seeds. So S, the phenotype S with unknown genotype, is crossed with WW. What is the role of the test cross? So it permits to identify the genotype of an individual with dominant phenotype, whether it is pure or hybrid. If the descendants are 100% smooth, then the tested individual would be pure, SS. If the descendants are 50% smooth, 50% wrinkled, the tested individual is hybrid, SW. As we said, if the test cross gives 100% descendants, then the tested individual is pure SS. Why? Because when SS is crossed with WW, both being pure, they produce only each one type of gamete, the first one producing only S and the second one producing only W, then the resulting individuals will be SW, 100% one phenotype S. However, in the second case, if the descendants are 50% S, 50% W, actually the individual will be SWY because SW crossed with WW, SW being hybrid produced half S, half W, while WW produces always one type of gamete, which is W. As a result, the descendants will be SW 50% phenotype S and WW 50% phenotype W. Let's move now to the second case of incomplete dominance or codominance. It's a cross between two parents of pure lines of opposite sex, where also one gene is studied, but in this case, the alleles of this gene are either codominant or incompletely dominant. What is incomplete dominance? It's when F1 individuals express a new phenotype that is intermediary between parental phenotypes then different from the parental phenotypes. Example, parents of red flower with white flower crossed together give in F1 only pink flowers. The phenotype pink is said to be intermediary between white and red, and thus we say the allele red and the allele white are incompletely dominant. In this case, both alleles are represented by capital letter. Now, what is codominance? It's when F1 individuals express also a new phenotype, but this phenotype is the expression of both parental phenotypes. For example, the first parent has the blood group A and the genotype AA. The second parent has the blood group B and the genotype BB. F1 will be of phenotype AB. It's not A, it's not B, but it's a combination of both phenotypes. Another example is when a white animal, for example, is crossed with a black animal. All F1 will be white striped with black. So white is not expressed alone in F1. Black is also not expressed alone in F1. However, both alleles, white and black, appear in the same individual F1. The phenotype white striped with black and the phenotype AB, they are the expression of both parental phenotypes. So we say allele A and allele B are codominant, or white and black are codominant. So let's do now an application concerning incomplete dominance. In 4-o'clock plants, the crossing of red and white flowered pure parents produce pink flowered plants. As you can see in this figure, red crossed with white produce in F1 only flowers that are pink. I want you please now to stop watching the video for a few minutes, copy these questions on a paper and try to answer these questions. So here is the answer of the exercise. For the first question, whether it's a case of incomplete dominance or codominance, it's clear here that it's a case of incomplete dominance. So the question is to specify whether it is incomplete or codominance, and when the question is specified, we know that we should answer and also justify our answer. So why it's incomplete dominance? Since the parents, red and white, when crossed together, they produce in F1 pink that is intermediary between red and white. Then for the second question, which is to represent the alleles by symbols, so the symbols are capital R for the color red and capital W for the color white, both represented in capital since it's not a case of dominance. For the third question, factorial analysis, the parents, red are R, white WW, parents being pure, each of them produces one type of gamete, then R and W. F1 will be RW 100% pink. Then, when we cross F1 individuals with each other, RW with RW, the result is F2 generation. F2 generation, as you see, is represented in the table of cross. So, the gametes RW 50% each, produced by the first parent, and RW 50% each, produced by the second parent. Thus, the descendants are RR 25%, RW 25, RW 25, and WW 25. So, as a result, 25% RR pure red flowers, 50% RW pink flowers hybrids, and 25% pure white flowers. You see, the self-cross in the case of incomplete dominance has given here three phenotypes, red, white, and pink, 25, 25, 50. While the self-cross in the case of dominance, like the case of smooth and wrinkled seeds, has given two phenotypes, 75% smooth and 25% wrinkled. Please do the following homework and thank you for your attention. See you later.